All right, so chapter 19 is all about viruses. So the virus that you just saw on that opening slide is known as a bacteriophage. It is able to infect and then take over bacteria. And in the case of the bacteria you saw on that first slide, that is an E. coli. Viruses themselves are not living things. They are able to take over life forms or utilize cells' metabolic machinery to be able to replicate themselves. And so these are actually, viruses are what we use to first better understand what was happening in cells, so where the origins of molecular biology came from. So what are viruses? They are nucleic acids surrounded by proteins. They were actually detected long before we could actually visibly see them. One example of this was with tobacco mosaic disease. This disease basically stunts the growth of those plants and leaves them with this mottled mosaic coloration on their leaves. And it was thought back in the late 1800s that perhaps there were some particles smaller than a bacterium that was the source of this disease. And Wendell Stanley was first able to confirm this hypothesis in 1935. He crystallized that particle and it is now known as the tobacco mosaic virus, TMV. And so there you can kind of see that process. Viruses, again, are not living things. They are not cells. They are super duper small. They have a nucleic acid that's enclosed in a protein coat, and then depending on the type of virus, that may be with surrounded by a membranous envelope. Viral genomes can be double-stranded or single-stranded DNA or RNA, and obviously the type of nucleic acid will determine the type of virus it is. That's like the T4 robot. So capsids are the protein shells that protect that viral genome. They are built from capsomeres, which are protein subunits that can come in all sorts of structures. And again, some viruses will have those envelopes that do provide them with an easier way to enter cells to infect their host. The viral envelopes are found around influenza capsids, along with many other animal viruses. These envelopes are developed in part from the host cell's membrane, but they do have viral molecules along with host cell molecules making them up. So here are just four different types of viruses. You can see the different shapes. The capsomere, that's a cylinder. The capsomere, that's with the tobacco mosaic virus. The adenovirus, which is like an icosahedral capsomere. You have the influenza virus, which has the capsid as well as the membranous envelope. And then you have the robot T4 bacteriophage that's got the capsid head. So you have all sorts of shapes, and when you see those glycoproteins on the outside of the surface of either the capsid or the membranous envelope, those are ways to help the virus find a way into living cells to be able to infect it. So bacteriophages, which are also called phages, are viruses that are able to infect bacteria. They have some pretty complex capsids. The phages have a capsid head that will enclose their DNA, and this is the type that's the robot type one. They have a tailpiece that will help to attach the phage to its host cell, and then it can use that to inject its DNA inside of it to take over it and to make more copies of that phage. Viruses, again, are not able to replicate independently. They are obligate intracellular parasites. They can only replicate within a host cell. There's only certain host cells they can infect. They do each have a host range. When they do enter, they are able to manufacture their proteins so that they can make more copies of the capsid. They're able to utilize the ribosomes to copy their DNA into proteins, and they're also able to use the various enzymes to replicate their DNA or their RNA. So as a part of those processes, they can use the tRNAs up, the amino acids, ATPs, and then they will exit the cells and spontaneously self-assemble into new viruses, or they can form them before they exit the cells. So there you see the entry. You see it taking over the host cell machinery and then producing new virus particles. Phages are the viruses that are best understood. They have two different reproductive mechanisms. They can just completely destroy the cell, and that's with the lytic cycle. And then they can also become embedded into that bacteria's DNA, and that would be the lysogenic cycle. The lytic cycle is going to again cause the death of your host cell. It produces lots of new phages and lyses the cell's wall. So this is just for bacteria, your prokaryotes, releasing these the virus progeny. When you only have the lytic cycle involved, these phages are known as virulent phages. Bacteria do have ways to protect themselves against phages. We're going to talk about this a little bit more in chapter 20 with biotechnology. They can use restriction enzymes that help to recognize and cut up certain sequences found on phage DNA. So there's your lytic cycle again, and the robot type virus, your T4 phage. The bacteria is the chromosome is injected. It breaks down the host cell's DNA. It then uses the host cell's ribosomes. Remember, this is bacteria, so we don't have a nucleus to copy its DNA and to make its proteins that it needs to form its head and its tail and its fibers. Uses up as much as it possibly can. Those get self-assembled and then released to go attack other bacterial cells. The lysogenic phase is able to replicate the phage's genome, but it doesn't destroy your host cell. It incorporates it into that host cell's chromosome, where it's now known as a prophage. So every time that cell undergoes division, it doesn't just copy its DNA, it also copies the phage's DNA. And there are environmental signals that can trigger the virus genome to leave that chromosome and to start to move into a lytic cycle where then it would go through what we just discussed, take over this host cell's machinery, copy its nucleic acids, make its proteins, make lots of progeny, and then destroy the cell and leave it. When you have phages that can utilize both the lytic and the lysogenic cycles, they are known as temperate phages. So there's the process of the lysogenic cycle. You can see the blue is the phage's DNA, and then the brown is the bacterial's chromosome. And over there on the top right-hand corner, you can see that the bacterial chromosome now has the phage's DNA incorporated in it, where now it's known as a prophage. Again, it has the option to replicate, or it has the option to exit and to go into the lytic cycle. We're going to talk about animal viruses now. These viruses are classified in part by the type of nucleic acid that is found in them. Are they DNA or RNA? And are those nucleic acids single-stranded or double-stranded? So there are lots of classes of animal viruses. We can have double-stranded DNA. We see those with adenovirus, papavirus, herpes virus, and pox virus. We can have single-stranded DNA, which is found with parvovirus. Double-stranded RNA is seen with reoviruses, and we can have single-stranded RNA. This is able to serve as directly as an mRNA template, so nothing else is needed. It can go straight to translation. Picornavirus, coronavirus, flavivirus, togavirus. Then you can have the single-stranded RNA that can be used to make the mRNA that then could be translated. The filovirus, orthomyxovirus, paromyxovirus, gosh, I can't say these names, rhabdovirus. And then we have single-stranded RNA, which actually works in reverse, goes against the central dogma, and that would be retrovirus. And that's what we see with RNA tumor viruses like leukemia, as well as with HIV. Coronavirus is going to be able to serve directly as an mRNA template. And then you also notice it had some where it talked about envelopes. So we have a couple of double-stranded DNAs that have envelopes, and we have several single-stranded RNAs that have envelopes as well. So there's a lot of viruses that infect animals that do have that envelope. And on the envelope are glycoproteins that are viral that can bind to certain receptor molecules on the host cell surface. And so when they are able to bind, they get formed. That helps them enter the cells. But these envelopes get formed from the host cell's plasma membrane. And so that may be part of where they're getting these from. And then you could have viral membranes form initially from the nuclear envelope and then get replaced by an envelope from the Golgi, from the processing portion of your eukaryotic cells. So here you have an envelope with glycoproteins. It's got an RNA inside of it. And then the glycoproteins help it to be able to find the correct key to enter into the cell through an appropriate receptor. The capsid and the RNA move in. The RNA gets used either directly to make your proteins in translation or to make a template. It then can get copied, and the capsid proteins as well as the glycoproteins can get made, and additional virus molecules are formed. So the cell itself remains intact, but it gets a lot of its resources get used to make additional copies of that virus. The RNA genomes are the ones that we find in quite a variety in these animal viruses. We talked about how retroviruses are a little bit unique in that they use reverse transcriptase to turn their RNA back into DNA, which is not the way things go. We take our DNA and we transcribe it into RNA. HIV is the retrovirus that is known to cause AIDS. And so here with HIV, you've got two identical strands of RNA. You've got your capsid protein protecting that nucleic acid along with the reverse transcriptase enzyme, your viral envelope, which is studded with those glycoproteins. The glycoproteins help it to enter the cell. Why that reverse transcriptase is so important, the viral RNA is then able to be turned into a hybrid of RNA and DNA. That DNA then can be just turned into double-stranded DNA and incorporated into a chromosome. We talked about prophages before with the bacteria. This would be a provirus. It can still make additional copies of that virus or the proteins and the enzymes and the envelope and the nucleic acids that would make up other virus molecules, but now you have the virus incorporated directly into the cell. So when it is replicated, that DNA from the virus will also be replicated, and then that cell can continue to make additional virus particles. So again, viruses don't fit our definition of living organisms because they cannot replicate independently. It is thought perhaps that they evolved from bits of cellular nucleic acids. Some sources of viral genomes are your plasmids, the circular DNA we find in bacteria and yeast, transposons, those are the mobile DNA segments, and these are all of them, the plasmids, the transposons, and the viruses are acting as mobile genetic elements. Mimivirus is the largest virus that has been discovered so far. It's a double-stranded DNA virus, and it's equivalent to the size of a small bacterial cell. And so there's some debate about whether this virus came about before or after cells. More to be determined. And so the final piece we're going to talk about are some other types of pathogens. We've spent some time talking about viruses and how they can have some significant impact, but so can viroids and prions. So viral infections can, as we are well aware, affect humans, agriculture, crops, and livestock. But viroids and prions can also cause some disease in plants and animals. Viroids for the plants, prions for the animals. So when we have viral diseases in animals, they can damage the cells. They can also kill them because they can result in the release of enzymes from lysosomes that can destroy things, your hydrolytic enzymes that are found there. They can also result in the cells producing materials that are toxic, and that could lead to disease symptoms. And then sometimes it's just the envelope proteins. And as we've seen with the coronavirus, some people have no reaction, and some people definitely have had very severe reactions, including losing their lives as a result of being affected with coronavirus. So vaccines, we're definitely learning a lot about vaccines these days, are derivatives that are harmless of pathogenic microbes that basically stimulate our immune cells, our immune system to come up with a defense to help prepare it to fight those pathogens. Um, they will only work, vaccines will, for viral illnesses. Viral infections cannot be treated with antibiotics. There's no cell wall, and that's typically what antibiotics are going to go after, is trying to come up with a way to break down those bacterial cells. Viruses are not cells, so that would not work very effectively for them. There are medications that can be given to treat symptoms associated with viral infections and to help to reduce viral infections, just the symptoms of them, but they will not cure the viral infections on their own. Okay. And so here in this particular example, you've got your nucleic protein, you've got your lipid envelope, and you see how you've got the neuraminidase and hemagglutinin. Those are the ways, those are the glycoproteins that are going to be used to help attach this virus to a cell's receptor. When the neuraminidase, I can't say that word, doesn't fit appropriately into a receptor, that hemagglutinin will come along and break it off so that it can try another receptor, so that it can move around that cell's membrane, trying to find a way to enter into the cell. And so that is one way that antiviral drugs might be able to work, is just to help to treat some of those symptoms that are going to come about from the impact of the viruses trying to attack your cells. Emerging viruses, ones that suddenly become apparent. When I covered this last year, COVID was out, but it definitely was not anywhere near where it is today. COVID would definitely be an emerging virus. We had an epidemic of a flu-like virus, nothing like COVID, when I first started teaching. That was known as H1N1. It was showed up in both Mexico and the US. It was known as a swine flu because it was a combination of the traditional virus, but that it had also had some of swine DNA making it up. These are epidemics caused by strains of influenza that we don't have immunity to, that our B cells and T cells have not developed the ability yet to recognize and to fight off. Pandemics, a viral disease that's found in a small isolated population. When it emerges, it can go global. All it takes is one person taking it somewhere. If you have not watched Contagion, make sure your parents are okay with you watching it, but it is a unfortunately very realistic look at what we've been going through over the past year. Viral diseases can emerge when they basically jump between animals and humans. When they jump species, they can exchange that genetic information, and that's going to cause new combinations of proteins to be formed that our systems don't recognize and we don't have immunity to, hence COVID-19. The swine flu, as I said, was initially thought to have been passed to humans from pigs, so it had some of that swine DNA embedded along with the regular virus, the regular influenza. So I said we were going to talk about some other things besides viruses. We're going to look at viral diseases in plants. These are known to cause discoloration, stunted growth, and damage to flowers or roots. The plant viruses, too, typically have an RNA genome. They can cause disease by either horizontal transmission entering through cell walls or by vertical transmission via a parent. We can also have viroids. Viroids are small circular RNA molecules that are able to infect plants, and they will disrupt their ability to grow. And then prions, we do not see in plants, we see those in animals. Mad cow disease and scrapie are some examples of where prions have come into play. They are slow acting, and they are virtually indestructible. So, when an animal is found to be afflicted with mad cow disease, there's not much that can be done other to than to put that animal down and any others that encountered that animal. These can cause brain diseases, and the concern obviously is that those prions then could be passed on to humans. They propagate by taking normal proteins and converting them into their prion form. And again, there's a couple of examples down there. Creutzfeldt-Jakob disease is another one that's caused by prions. So there's your prion, again, a totally different shape from the normal protein. It will change the shape of those normal proteins, convert it to be a prion. So now you have stacks of these prions and eventually aggregates of them. And that's going to lead to the plaques that we see with individuals afflicted with diseases from prions or even animals in general.